Hi, this is Miss Hale, and this is your hair notes for forensic science. Remember, our central question is how hair and fiber evidence can implicate a suspect at a crime scene. And right now we're working on the hair part of our unit. So we're looking at how hairs found at a crime scene can point to a specific individual. So the function of hair, there's three major functions of hair. It regulates your body temperature. It decreases friction, so if things rub against your arms, they're going to rub against the hair first. And it also protects against sunlight, so you can think that it's on the top of your head, and it's actually protecting the top of your head from sunlight. That's why bald men often get sunburns on the tops of their heads. So the structure of the hair we've been talking about in class as well. It grows out of a follicle in your skin, and that produces the hair shaft, which is the part of the hair that's coming out from your skin. And there's three layers. They're illustrated up here. So we have our medulla, which is in the middle, the cortex, which is the bulk of the hair and contains the pigment, and the cuticle, which is those imbricate, spinous, and coronal parts of the hair. And so that is kind of the scaly part of the hair on the outside portion. So here's a structure of the hair within the skin. You can see the follicle here, some glands that produce oil for the hair, and then the hair shaft that comes out. Remember that the DNA is found in this part, the follicle of the hair. So if you think of the hair as a pencil, or similar to a pencil, you can think of the paint on the outside of the pencil as the cuticle, the wooden part of the pencil as the cortex, and the inside pencil, the medulla. And this is the picture here, really, that you should be drawing of the three layers. So there's different types of cuticles and cortex. The cuticle, again, is the outermost layer of the hair, and it has overlapping scales that protect the inner layers. We've talked about the three types. Oops, sorry. Oh my gosh. Okay, sorry about that. It has overlapping scales, and there's three types: coronal, spinous, and then imbricate is the kind that you find in human hair. The cortex is the thickest layer. It contains most of the pigment of the hair, but the distribution of the pigment varies. It's usually denser near the cuticle with humans, but oftentimes in animals you'll find that the pigment is denser near the medulla. Here's our different types of medullas. This is talking again about human medullas. So these are all amorphous. You can see that we have continuous, interrupted or intermittent, fragmented or segmented, a solid medulla, which is the whole hair looks to be the same color, and then none, where the medulla is just absent. And here's some different types of hair. So you can have buckled hair, where the hair looks like it kind of has a split in the middle, so it's like this. Blunt hair, and a double medulla, where you actually see two different medullas in the same hair. You can look at a cross-section of hair, so meaning if this was the hair, you're cutting it this way across. And that can be circular, triangular, irregular, or flattened. The shape of a cross-section is going to influence how curly or straight the hair is, and the texture, whether it's coarse or fine. Hair has three stages in its life cycle. It is the anagen stage, where the hair is actively growing. That's the first stage. And so the cells in the follicle are going to rapidly divide. Remember, that's the part in the skin where the hair grows from. And they'll deposit materials in the hair. So if you are doing drugs, that will get deposited in your hair that way. Then the second stage is the catagen stage, where the hair grows and changes, but it's not growing quite as rapidly. And then finally the telogen stage, where the follicle becomes dormant, and then the hair dies and oftentimes will fall out. Treated hair, you can tell when you look at a crime scene whether hair has been treated if you look at it under a microscope. You can tell if the hair has been bleached because it disturbs the scales on the cuticle, it removes the pigment from the cortex, and it oftentimes leaves the hair brittle and it looks very yellow. Whereas dyeing, which is slightly different, will color both the cuticle and the cortex, whatever color it is. So you can tell how recently hair has been dyed, which would help implicate a certain person, perhaps. There's also racial differences among hair. Broad racial groups do exhibit some shared physical characteristics, but it's really not applicable to all individuals in these groups. Therefore, we can't definitively assign a hair to any specific racial group. So some differences between animal and human hair. First is pigmentation. Animal hair has denser pigments towards the medulla, where human hair tends to be more dense towards the cuticle. You can have banded color patterns where there's different colors throughout one length of hair. That's possible in animals, but really not in humans. And the medulla is much thicker in animals. The medulla makes up more than half of the hair, while in humans, the medulla is only a third or less. And so you see a picture of that here. So we can see that the medulla makes up more than half the hair in cattle, but in human it's less than 1/3. In animals, cuticle scales resembles petals, which you can see here with the spinous cuticle or a stack of crowns. But in humans, it's commonly flattened or narrow, and we call that an imbricate scale pattern. When we're using hair in an investigation, we can do a macroscopic investigation, meaning looking at it under the naked eye or with a hand lens, and we can indicate the length, color, and curliness of the hair, and that could limit the group of individuals that left that hair at the crime scene. You can use something called phase contrast microscopy, which will show you whether the hair has been dyed or treated. Could be also using a perm or something like that. And if you use an electron microscope, you can have even more detail about the hair. An electron microscope will look at the hair, the atoms in the hair, rather than the hair structure in general. So this is a microscopic image from an electron microscope, and you can notice how you can really see the scales of the cuticle and the pigment granules in the cortex really well as compared to what you guys saw in your microscope lab. Other tests you can do on the hair shaft include chemical tests, so you can look for different drugs that were in the hair. Yes, they can take your hair and do drug testing on it. You can also look for exposure to toxins, so if you think that the person has been poisoned over a certain period of time, you can look for those poisons in the hair shaft. And you might do something called neutron activation analysis, and so you would use that if you think that there's a chemical in the hair to determine what chemical is there and how much of it exists. So when you're testing a hair follicle, obviously a microscopic assessment is going to be really cheap and quick to do, so you can just pop it right under the microscope and look at it that way. A lot of times they'll have comparison microscopes where you can look at two samples at the same time. You might be able to determine blood type through a hair follicle if you have the root of the hair. So this is if the hair has been pulled out. And then you can also do DNA analysis, and so that's going to be the best method to use to identify a specific individual. You can do mitochondrial DNA analysis if you have just part of the hair, but if you have the root, if you have the follicle, you can do regular DNA analysis, which is quite useful. And then we can narrow it down to a specific person. And the last thing is that when you're preparing a microscope slide with hair, the easiest way to do it is to take your glass slide, put your hair right on the top. Notice that this has both the end of the hair and the root of the hair, and it's just taped right on. There are other ways to do it that are a little bit more involved, but they are more expensive, and oftentimes this is the best way to do it in a quick and cheap way. So those are our hair notes. Remember to think about how these will help you implicate a specific person if you were to find hair at a crime scene. Please write down any questions that you have and make sure you bring them to class with you next time. Thanks, guys.